So I'm very grateful for the organizers of this meeting for giving me the opportunity to present the topic, and I think it's very relevant to HIV field. And I also felt from the presentations that we heard today and yesterday that this field was not really dealt with at all. So we all know that drug resistance is quite an actual problem in the treatment of HIV. So the question is that right now we have very strong, well-adapted treatments constituted of several types of drugs against different viral targets. And in most of the individuals, they give a good response, decreasing the virus load to almost undetectable levels, making these individuals not dangerous to the surrounding people due to the very low viral load. And then the cost of the treatment also went down. And of course, there are backsides to the treatment, as people have to take these drugs every day, and it's really a problem. But still, it could almost be considered as a chronic disease, as other chronic diseases, lifelong chronic disease. But we have a phenomenon of emergence of drug resistance. And also, as was described recently, that drug-resistant strains, which then circulate in quite high levels in individuals, could be transmitted to healthy people and then cause primary drug resistance in these individuals with the early failure of treatment for these individuals on the different HAART regimens. Also, drug resistance could multiply in individuals from the risk groups that are not adhering to treatment regimens, and also still they could expand in the groups where not adequate HAART regimens are applied, which miss few drugs, or when drugs are exchanged one for another one, leading to more and more resistance. And it has been assumed that in a few years from now, the rate of infection with drug-resistant strains could be over 15% and approaching 20%. And what could we do about that? Of course, we can invent new drugs, but then I would say it's like a dead race almost. On the other hand, what we know that there are special cohorts of HIV-infected individuals. They are elite controllers, that people who can control to a certain extent the viral replication, and in certain individuals it can continue for very long periods of time, so, which would mean that in principle an immune response on a certain, probably certain genetic background could help very much to the suppression of the virus replication. But what happens when we treat individuals with HAART is that they lose antiviral response because viral circulation is very low and it doesn't give any impetus to the immune system. And that has been addressed by so-called structured therapy interruptions, when they tried to give a virus a chance to produce more antigen and boost immune response, but that approach was sort of banned. It was considered to be really bad because it increases integrated virus load, and it's no good. So we have to look elsewhere for the stimulation for immune system, and I think that immunization, specifically genetic immunization, can be one of the means to keep the memory of the organism to the virus while a person is on HAART. So this is not a vaccination in the sense of prevention primary infection. It's more like a supplement to therapy, or one can also think of it as a preventive vaccination if you hopefully in the perspective of the chief prevention of infection with drug-resistant strains. So our aim is to try to prevent the emergence of the primary mutations of drug resistance to create immune pressure on the virus that will act prior to HAART or in parallel to HAART, and create some sort of a bottleneck effect, preventing virus to mutate towards drug resistant, or at least hindering this process, making it slower and less efficient. And we chose naked DNA as this immunotherapy or vaccine vehicle, and a simple plasmid backbone. And we took into consideration the main proteins responsible for the drug resistance, HIV enzymes, the first players, so to say, in the viral replication. And also because they are the targets of the cellular immune response, much less extent antibody response, and then the gene vaccines, which are known to be very strong in inducing T-cell response, would be very applicable for that. So what are the design principles? We use the same principle as used by a group of David Weiner. It's a consensus sequence, and it was sort of supported by the fact that in the large territories of Central Europe and East Europe and Asia, there is one clade A strain, which is dominant. It is infecting more than 80% individuals. It's so-called EDU, intravenous drug user strain clade A, or former Soviet Union strain clade A. And it's very homogeneous because for a long period of time it was transferred between intravenous drug users with a very short time in between primary infections, and the virus did not really mutate to a large extent in between the infections. And on top of this consensus sequence, we introduce primary drug resistance mutations to the drugs abundant and in use on that territory. And of course, as a safety feature, it's an activation of enzyme activity, because none of these enzymatic activities, like reverse transcription or integration, is something that we want to have in the cells of the vaccinated individuals. And the history of how the creation of these genetic vaccines would go would be described in the talk on the next session given by Dr. Starodubova. But I just show one slide where it was started like 15 years ago with wild-type reverse transcriptase, then was expression optimized, then it was optimized and inactivated. Then it was a wild virus gene, like with drug-resistant version RT114, and again, which we optimized and inactivated. So it's just one, and then published in this series of papers during these years. And we did the same for HIV integrase, but then we went straight to synthetic expression optimized genes, and did not work with the viral genes at all. And here I will tell you the story that we have not yet published, but we've been working extensively on that. It's HIV protease, which is very important for the virus because it is responsible for maturation of the polyprotein, and it's one of the major targets of antiviral therapy. And a lot of drug-resistant mutations, which are given here in red, emerge quite soon, specifically if individual is taking just anti-protease inhibitors. So what we did, we took two selections of HIV sequences from the territory of former Soviet Union. One, a larger one, was set of sequences from middle of 90s to 2000. And the other one was sort of younger, ending up at now. And we analyzed the sequences and created the consensus on the basis of the first and the second sets. And to our surprise, although we expected it, but we were really surprised to see that those consensuses were totally identical, meaning that in that protease is very conservative, specifically certain motifs, but the protein has not changed to a large extent during quite a long period of evolution, which would mean that genetic vaccine or immunotherapeutical created on the basis of this consensus would be effective for yet some years to come, thinking of the evolution rate. Then we analyzed the mutations that confer resistance to protease inhibitors, made the analysis of studies with different inhibitors, and selected the three most common primary mutations typical to clade A strains. And we created synthetic genes, wild-type genes, and genes with inserted mutations of primary resistance. And we faced the problem of non-recognition of the recombinant proteins by the available anti-protease antibodies of clade B. So then we used a different approach. We didn't work in transfected cells to confirm the expression, but we injected the genes into the oocytes of the African frog Xenopus laevis, and we made the metabolic labeling. So we added 14-carbon lysine, and then we measured the amount of newly formed protein of the correct molecular weight, and compared the new genes in the efficacy of transcription to the already characterized gene of protease clade B. It's a dark bar in the corner. And we found that genes for protease with inactivation mutation were well expressed in eukaryotic cells. So then it was a time to put them into genetic immunization. Here is the simple first round set of immunization experiments in mice, where we did a single immunization with 20 micrograms of the gene at two sites, and then in continuation, the genes that were found to be effective were subjected to also boosting experiments. And we assessed immune response by ELISA, ELISpot, Fluorospot, and flow cytometry assays for multiple cytokine secretion. And what was very important on all these studies that one has to follow an optimized protocol for the vaccine delivery. So we use needle injection, which is commonly used, and optimize that. And then we, of course, use the electroporation over the injection sites, as we heard today. Very strong presentation from Inovio showing how good it is to electroporate. So we also use that. And the injections we need by needles, and we also tested microneedles, and we tested a bioinjector. And the microneedles gave a very good result, although it's sort of expensive in preclinical even trial because of the cost of the needles. And these optimizations were described in a series of publications given below. And we were able to monitor the efficacy of gene transfer by by in vivo imaging at the presentation that will be given after my presentation, so I will not go into any detail except for showing that you can see where you have injected your gene, how well it is expressed, so how well you delivered, so that you would not blame your immunogen for poor performance if it was delivered in a poor way. So now we know that we deliver in an optimized fashion. And then you know also where the immunogen is expressed, because you can localize the area of expression. You see it on three-dimensional figure of the mouse body. You can see it's expressed at a certain distance from the surface, which would be this muscle injection. And you can also see similar expressions on the skin if it's an intradermal injection. And when these optimized gene immunogens were delivered in an optimized way with a follow-up of expression at the site of injection, so we can demonstrate that these synthetic gene immunogens actually perform very well. And here you can see the interferon gamma and IL-2 Fluorospot results, where the protease consensus clade A gene with inactivation, which is a pink bar, is compared to protease clade B viral strain optimized for expression. This is a gray bar. You see that shift from viral gene, even expression optimized, into consensus clade A, in this case, sequence give a several-fold increase in cellular responses in terms of interferon gamma and IL-2 production. And here is the overall integrated result of our studies. So we have characterized in here a dual interferon gamma L2 responses to reverse transcriptase, to protease, to integrase of HIV clade A and clade B also on the basis of HXB2 strain. And also the humoral immune responses induced by intradermal and intermuscular injections of these genes. And in some cases, as for RT, we were able also to detect quite strong mucosal IgA response. And these studies are published. And summary of what we offer to develop for therapeutic use are the plasmids encoding HIV enzymes, with primary mutations of drug resistance, with no enzymatic activity on the background of clade A and clade B strains, specific for East Central Europe and Asia. But as it was already repeatedly mentioned today, that the DNA vaccine platform allow a rapid shift from one immunogen to another, also like with the synthesis of the genes or with insertion of point mutations. So this principle could be applied for design of new immunogens that would be ready to use within the 2 months from the onstart of the project. Also, we have the effective protocols for the delivery of these genes, and we have shown that all these components induce strong polyfunctional response in rodents, and we start now rapid immunizations with the genes. And what we think could be done in perspective using this platform, it's the HIV sort of vaccine or immunotherapy trial in which you could immunize individuals either prior to HAART or in parallel to HAART. And in contrary to what was done before, when to evaluate the vaccine efficacy, you have to stop the drug treatment and then see if your immune response can restrict the virus load. And that was no good because in most of the cases it couldn't. And then again, the patient is suffering from excessive viral replication and integration, and therefore it starts to be kind of difficult to recruit individuals for that sort of vaccine trials. So our protocol would not include any STIs nor unsafe procedures in testing. It would be a combination of HAART with immunization, and the efficacy measurement would be the frequency of drug-resistant mutations in this cohort of individuals as compared to individuals on HAART without any immunotherapy. And here I would like to bring my acknowledgements to the people from 3 different entities working on the project, 2 in Moscow and 1 in the Karolinska Institute, and to the grants from different sources, Swedish, Russian, and European. And now we are like the ladies fishing, expecting to get our golden fish. So, and I would very much appreciate any comments and questions and suggestions from you. Thank you very much.
For discussion. Are there any questions? Single question. No questions? The organizer gives you a certificate of appreciation.
Thank you very much.
Congratulations.